Hello, and welcome back. We're going to kind of wrap up transcription here with this video. There's one more thing that happens in the process of transcription that I haven't talked to you about yet, and that is a process called splicing, okay? And like its name kind of sounds like, if you've ever done any electrical work or if you've watched electrical work done, right, the electricians will splice wires together, meaning they take a wire from one place and another place and they connect them, okay? That's what we're going to have happen with transcription also. And the process of transcription is going to happen in the nucleus. And before that transcript, okay, in this case, our messenger RNA can leave the nucleus. We have to modify that RNA that comes off of our DNA template, okay? So our messenger RNA is going to be composed of, and let me get two different colors here. I tend to draw them as boxes and lines, just so that you can distinguish them. But really what you would have here within this box, you would have some RNA sequence, some AUGCC, okay? You would also have some sequence that makes up the wavy lines in between, okay? But the way genes are organized in eukaryotes, and this is going to be important because this is going to be primarily a eukaryotic function. The way those genes are organized are going to be exons and introns, okay? So my boxes here are going to represent my exons. And in this case, this particular gene, okay, has 3 exons. The number of exons and introns is variable. The yellow squigglies are going to be the introns. And what's going to happen with the process of splicing is you're going to take the exons, woohoo, nice and squeaky, and you are going to splice them together. Because the exons are going to be what contain what we call the coding information, and we'll talk more about coding when we talk about translation. But essentially what I mean when I say coding is this is going to be the RNA sequence that makes our protein, okay? The introns, the sequence in the introns, does not make a protein, typically, okay? And it used to be that the introns, when I was in school way long ago in the dark ages, it used to be that introns were considered junk. But it turns out those introns, those small bits of RNA that get spliced out, actually are not junk, okay? Those small RNAs can be used to regulate cell processes. And that's important because when we're talking about the proportion of your DNA that makes up exons, okay? Approximately 2% of your genome, your DNA, is going to exist as exons, okay? And when I was in school, the way that was presented was the other 98% of your DNA is junk. And if you think about it, it's not very efficient to have 98% of something be junk, all right? So while it is only about 2% of your genetic material, that will code for protein, okay? And by protein, I mean the exons here, okay? The other 98% is not junk, okay? That other 98% is going to form things like your tRNA, your ribosomal RNA, and your small RNAs that actually function in regulating cellular processes within the cell, okay? Don't worry about what processes it's regulating just yet. When we get to the chapter, which is going to be chapter 11 in your textbook, about gene regulation, we'll talk a little bit more about how those small RNAs regulate some things. But for right now, just know that that's where that other 98% falls into, okay? So splicing, we're talking about taking exons and removing the intron sequences, okay? Now within that splicing, you can have things that will splice exon 1, 2, and 3 together, okay? And the way that your cell is more efficient about regulating the information is you can have some alternative splicing that happens, okay? So it might be that the majority of this particular gene that I have here is going to splice exon 1 to exon 2 to exon 3, okay? But it may be able to make another protein from that sequence if it splices exon 2, ooh, squeaky, exon 1 directly to exon 3. Okay, that would make a different protein. And that's a way that the cell can kind of get the most out of only having 2% of the genome be the RNA that will code for protein, okay? There can be some alternative splicing that happens there, okay? And in this case, the alternative splice would be to essentially remove exon 2, okay? It's going to remove all of that intervening sequence and directly splice exon 1 to exon 3. Okay, in terms of that, that's going to be the process of splicing, okay? There are also going to be two more things real quick that I'm going to add here that tend to be added to RNA before it leaves the nucleus. At our 5' end, there's going to be a cap that's added. That cap essentially protects the RNA from degradation out in the nucleus, okay? The other thing that's added to messenger RNA at the end is going to be a string of adenines. Oops, squeakiness. That string is called a poly-A tail. Okay, the poly-A tail is always added to the 3' end. That's a way for the components out in the cytoplasm to tell which is the 5' end, which is the 3' end of our messenger RNA molecule when it goes to make protein. Okay, that poly-A tail signals the end of it. The cap is going to be added to the 5' end, all right? Both sequences kind of act to protect the exons in there because you don't want to lose any nucleotides in your exons because then you won't get the right protein made, okay? So poly A tail and the cap, those are going to act to protect it. Those are also added to the RNA after it's been spliced, but before it leaves the nucleus. Once it's got the 5' cap and the 3' poly A tail, our messenger RNA now, okay, because what I've got here kind of in the middle of all of this mess, this is our messenger RNA, okay? It has the cap, it's got the coding sequence with the exons, and it's got the 3' poly A tail, all right? If you have any questions, please let me know.